Good morning, everyone. This is Rick Wojciech, or as they would say in Poland, Rick Wojciech. I want to welcome you all to the 2023 Rod Bell Memorial Lecture. So I want to start by giving thanks to everyone who made this event possible, especially Dr. Natalie Shaw, who took the lead to invite today's speaker and who will do the formal introduction. But before we start, I want to remind everyone that the Martin Rod Bell Memorial Lecture honors the memory of a scientist who is one of the founding members of NIHS. And I think, as we all realize, he was an unwavering advocate for the study of how the environment influences disease. So Dr. Rod Bell was the scientific director of NIHS from 1985 to 1989. And he was the chief of the signal transduction section from 1989 until his retirement in 1994. So this lecture series was initiated by scientists and friends of Dr. Rod Bell, and the purpose was to honor his many contributions to science and to the Institute. So Dr. Rod Bell, I suspect, as all of you know, he received the Nobel Prize in Physiology and Medicine with Alfred Gilman. That was back in 1994 for his discovery of the importance of guanine nucleotides in the transduction of hormone signaling on adipocytes. So Rod Bell made the discovery while working at the National Institute of Arthritis and Metabolic Diseases, or NIAMS, which he had actually joined back in 1961. So this discovery really provided the first biochemical evidence for the GTP binding proteins, or those molecules we call G protein coupled receptors. And these G proteins really transfuse hormone binding to receptors on the surface of the cell and into the production of second messengers within the cell. So G protein coupled receptor proteins have turned out to be one of the largest eukaryotic gene families, and today more than half of the drugs used clinically to treat human diseases target G protein mediated signal transduction. So this year's Rod Bell lecturer, Dr. Joanna Wysocka, who is the Laurie Lokey Professor in the Department of Chemical and Systems Biology and the Department of Developmental Biology at Stanford University. Her work on gene regulatory mechanisms underlying stem cell function, cellular plasticity, and differentiation is perfectly aligned with the theme of the Rod Bell Memorial Lecture. So her lecture today is entitled Making Faces, Transcriptional Enhancers in Human Development, Disease, and Evolution. And her expertise in the field of gene regulation and the developmental biology has earned her numerous accolades and awards throughout her career. So to learn more about these accomplishments and to formally introduce Dr. Wysocka, let me turn the virtual podium over to Dr. Natalie Shaw. Natalie, the podium is yours.
Okay. Thank you. I'll try to pronounce it as well as you did, Rick. So Joanna Wysocka, as you said, is the Laurie Lokey Professor in the Department of Chemical and Systems Biology and the Department of Developmental Biology at Stanford. She's also a member of the Stanford Institute for Stem Cell Biology and Regenerative Medicine and a Howard Hughes investigator. Dr. Wysocka is originally from Poland and graduated from the University of Warsaw in 1998. She did her graduate studies at Cold Spring Harbor Laboratory with Dr. Winship Her and postdoctoral training at the Rockefeller University with Dr. David Alice. She joined the Stanford University faculty in 2006 and less than 10 years later became a full professor and a Howard Hughes investigator. She is a recipient of numerous awards, including the Searle Scholar Award, W.M. Keck Foundation Distinguished Young Scholar Award, International Society for Stem Cell Research Outstanding Young Investigator and Momentum Awards, and the Vilsack Prize for Creative Promise in Biomedical Science, a prize that recognizes immigrant scientists who've made a major contribution to U.S. society. She was elected to the American Academy of Arts and Sciences in 2018 as an EMBO International member in 2019. She is internationally recognized for her work on gene regulatory mechanisms underlying stem cell function, cellular plasticity, and differentiation. Some of her group's most notable contributions include discovery of chromatin signatures associated with active and primed enhancer elements, development of new tools to visualize and study regulatory regions in living cells, and insights into the mechanisms by which changes in gene expression drive evolution, individual variation, and congenital craniofacial disorders. Thank you again for visiting with us today, and we look forward to hearing your talk.
Thank you very much, and it's really an honor to be giving this lecture, and big thanks for Natalie for inviting me, and I apologize I cannot be here in person today. Do you see my slides? All right.
Yes.
Beautiful. All right, so let's see. Very broadly, my lab is interested in understanding how biological information is integrated on chromatin. So in other words, how regulatory information that is encoded in our DNA is read in the context of the cellular history by lineage-specific transcription factors, and in the context of the signaling environment of the cell by signaling effectors of major signaling pathways. And we're really interested in understanding how these three layers of information encoded by the DNA, cellular history, and environment work together to then produce permissive or restrictive chromatin states, which in turn allow for precise spatiotemporal control of gene expression in development and in tissue homeostasis. So as many of you know, much of this information integration on chromatin occurs at cis-regulatory elements such as promoters, enhancers, silencers, and boundary elements. And of those, one class of cis-regulatory elements called enhancers plays truly a privileged role in mediating cell type specific gene expression programs. So enhancers are these modular, short genetic, non-coding genetic sequences that are able to activate expression of their target genes over large distances, and they're often specialized to direct expression in a specific spatiotemporal context. You probably also realize that from recent explosion of human population genetic data, we learned that most human phenotypic variation, as well as complex disease-associated genetic variants, map to the non-coding parts of the human genome. But within this non-coding genome, these variants are enriched within cis-regulatory elements, and especially within tissue-specific enhancers. So in my lab, we're interested both in fundamental mechanisms of long-range gene regulation by enhancers and other cis-regulatory elements, but we also want to understand how variation in enhancer sequence or function can give rise to the human phenotypic variation between species, or what makes us human, within the human species, what makes us individual, and how mutations in regulatory elements can result in disease. So we use multiple models in our studies, but one of our favorite model systems to study how cis-regulatory elements encode phenotypes is human face. So first, from the evolutionary perspective, recent changes in the craniofacial complex facilitated emergence of uniquely human facial features and a number of biological adaptations. And within humans, the face, of course, plays key roles in individual identity and in social interactions, and is a strong target for sexual selection. And finally, craniofacial anomalies account for a third of all human congenital diseases. And given the central role of the face in physical identity, these diseases have not only huge medical implications, but also devastating social consequences for affected patients and their families. So we recently became interested in exploring whether there are common molecular pathways that are associated with craniofacial disease and with a normal range facial variation that makes us all individual and look distinct from one another. So let me tell you a little bit about what we know about human facial genetics. So there are over 500 craniofacial syndromes, so really a staggering number, with Mendelian inheritance and manifestations in the face. So while these diseases are individually rare, collectively, they represent some of the most common human congenital malformations. And genetic mutations underlying these diseases are typically coding in nature and show either recessive or dominant inheritance. But interestingly, many craniofacial syndromes are actually associated with heterozygous mutations that are loss of function mutations in genes that are essential for embryonic development. So this suggests a very prominent role for haploinsufficiencies in craniofacial disease. So in contrast, normal range facial variation is associated with non-coding genetic variants that are enriched in enhancers that are active in facial progenitor cells called cranial crest cells. And to date, through GWAS studies, over 300 loci have been associated with normal range facial variation in diverse human populations. So not surprisingly, this is a highly heritable and highly polygenic trait. So we asked a simple question. What happens if we identify candidate target genes of these GWAS variants associated with normal range facial shape, and then intersect them with genes that are mutated in Mendelian disease? And the question was, will we see any common pathways appearing? And to our surprise, there was only one biological category that came up as enriched. And these are DNA binding transcription factors. So this, in fact, suggests that change in transcription factor dosage is a prevalent mechanism that is underlying facial variation in health and disease. So in syndromes, these transcription factor mutations are often indicative of haploinsufficiencies, which are predicted to result in 50% change in transcription factor dosage. And in contrast, non-coding variants associated with facial shape likely result even in much more subtle changes in gene dosage of much less than 50%. And it's a dirty little secret in the GWAS field that this variants, cis-regulatory variants associated with different traits only cause subtle 10 to 15% changes in gene expression. So we became interested in exploring how these relatively subtle changes in transcription factor levels influence phenotypes and determine their specificity. So one of these dosage-sensitive transcription factors in the face is SOX9. And in the context of craniofacial development, it plays multiple roles. It's involved in specification of this facial progenitor, key facial progenitor cells called cranial crest cells. It's required for entry of cranial crest cells into chondrogenesis program. And finally, it's also considered a master regulator of cartilage formation. And over the last few years, we've been using SOX9, both locus and the protein itself, to study multiple questions, such as role of non-coding mutations in disease and their role in regulating transcription factor dosage, transcriptional regulation of ultra-long ranges, and mechanisms underlying transcription factor dosage effects on phenotypic variation. And these three themes is what I'm going to focus on today in my talk. So SOX9 is developmentally important transcription factor that plays a multiple role beyond the face, and its haploinsufficiency causes a multisystemic disorder called campomelic dysplasia, characterized by limb defects, craniofacial defects, and male to female sex reversal. And what is really interesting from the regulatory standpoint is that the SOX9 gene sits in this enormous 2.5 megabases large gene desert. And the distinct non-coding mutations in this gene desert recapitulate some, but not other aspects of campomelic dysplasia. So for example, mutations in this region of the gene desert are associated with sex reversal in the absence of other manifestations. And mutations at the very end of gene desert are associated with isolated craniofacial defects. And these defects are referred to as the Pierre-Robin sequence, with the primary defect being underdeveloped lower jaw, which then in turn or in sequence leads to a number of secondary manifestations. So previously, Hannah Long in my lab identified 3 clusters of enhancers that are located at the centromeric end of the gene desert and overlapping at least one patient mutation of the patient's deletions identified in peer-event sequence. And we named these 3 clusters after the distance from the SOX9 promoter. So EC1.45 is located 1.45 megabases away from SOX9 promoter, and so on. And Hannah demonstrated that PRS locus enhancers are active during specific developmental window in this progenitor cells, cranial crest cells, and are later on decommissioned as these cells go through the chondrogenesis. So then she designed heterozygous deletions of each of these 3 clusters in human embryonic stem cells, then differentiated these cells to cranial crest cells and measured SOX9 expression in an allelic specific manner. And through this work, she showed that each of these elements contributes to SOX9 expression in cranial crest cells, with EC1.45 showing the highest contribution of about 50%, and the E1.35 and 25 showing smaller contributions of about 20% to SOX9 expression on the affected allele. So you may say, well, these are relatively modest reductions, but through in vivo mouse work that I won't have time to show you today, Hannah actually showed that craniofacial development is extremely sensitive to SOX9 dosage, and that even 10 to 15% reduction in SOX9 dosage during craniofacial development is sufficient to result in subtle but reproducible changes in lower jaw shape. So at this locus, even small perturbations in expression actually matter for the phenotype. But there were a few things in Hannah's paper that we could not really easily explain. And one of them was this mystery of the E1.35 element. So I've shown you that this element has marks that are characteristic of active enhancers. So histone marks and P300 association that are typically linked to active enhancers. And I also showed you that when we delete this element, it actually contributes to SOX9 regulation. And yet, when we tested the activity of E1.35 in enhancer reporter assays, it lacked autonomous enhancer activity. And this was in contrast to the other 2 elements, which were very strong and beautiful craniofacial enhancers. And this was not due to a species barrier, because when we tested these 3 elements in luciferase reporter assays in human CNCCs, we got essentially the same result. So this raises a question: how does this mysterious E1.35 element regulate SOX9 expression if it's not an enhancer? And more broadly, how do PRS locus enhancers achieve this very robust and precise SOX9 regulation over such long genomic distances of well over a megabase? So we hypothesized that perhaps a clue to this regulation may lie in the SOX9 locus topology. And to explore this, Hannah teamed up with another postdoc in the lab, Liang-Fu Chen, and together they worked with a fantastic colleague at Stanford, Alistair Buttiker. So Alistair's lab developed a DNA fish-based method called optical reconstruction of chromatin architecture, or ORCA. And this method allows for tracing chromatin fibers in single cells. So briefly, the region of interest can be sequentially imaged with probes that are read out by different barcodes. And then from the sequential imaging, you can reconstruct 3D chromatin traces, and from hundreds of thousands of such traces, then ensemble average maps can be derived. And those ensemble average maps resemble those high C maps that by now everyone in chromatin field is quite used to. So then to image SOX9 locus topology, we design a series of probes styling the 4 megabases of the DNA covering the entire SOX9 topologically associated domain, which is one of the largest TADs in the human genome, as well as neighboring TADs. And we had barcodes corresponding to each of the PRS locus elements, as well as to the SOX9 promoter. And then we image SOX9 locus topology in two distinct cellular states: in human embryonic stem cells, in which PRS locus enhancers are inactive and SOX9 is fully transcribed, and in CNCCs, where both PRS locus enhancers and the gene are highly active. And from over 1,000 single-cell, single-allele chromatin traces in each cell types, we then reconstructed and sample average contact frequency maps. So it was immediately clear that there are pronounced changes in locus topology between human embryonic stem cells and CNCCs. And in particular, in human embryonic stem cells, the smaller blocks of self-interacting chromatin reminiscent of previously described subtop structures were observed. And in contrast, in CNCCs, the most distinct feature of the contact map was the presence of these two internal architectural stripes, one corresponding in position to the promoter and the other to our mysterious E1.35 element. And other than those distinct architectural stripes, the interactions across the entire TAD are much more uniform in CNCC as compared to human embryonic stem cells. So then you can look at subtraction maps of these contact maps to see what interactions are most changed between the two cell types. And when we've done this, we saw that the most pronounced differences between contact maps in CNCC and human embryonic stem cells were increased contacts between the promoter and the PRS locus, so harboring all three PRS elements, and shorter range interactions were more prevalent so more promoter proximal interactions were more prevalent in human embryonic stem cells. So then we asked, how does distinct cell type specific architectures arise at the population level? So we considered two plausible hypotheses. The first hypothesis is that arise through the presence of chromatin conformations that are unique to each cell type. So each cell type has sort of their own unique topology. But the second hypothesis or possibility is that these differences in the average maps arise through cell type specific changes in frequency of conformations that are present in both cell types or commonly sampled by both cell types. And the single chromatin fiber nature of our data allowed us to distinguish between those two possibilities. So first, we used t-SNE analysis of chromatin fiber traces to see if we can detect any cell type specific clusters. So we focused initially on pairwise distances of all versus all positions within the entire SOX9 topologically associated domains. But as you can see from here on the t-SNE plots, we did not observe clear separations of human embryonic stem cells and CNCC conformation. So you see this salt and pepper pattern without any distinct cell type specific clusters. So then we reasoned, well, perhaps since in the average maps, we see that the largest cell type specific differences in contacts are observed between the promoter and these different regions of the locus, in particular, the very distal PRS locus. We then reduced our analysis to all pairwise distances of the SOX9 promoter to the remainder, all other points within the topologically associated domain. But again, we observed no clear separation, although now traces from human embryonic stem cells were biased towards one region of the t-SNE plot, and traces from CNCCs were biased towards the other area of the plot, suggesting that perhaps there are no separate clusters, but there are different frequencies of certain conformations in these two cell types. So to explore this further, we performed k-means clustering of the SOX9 locus topology, identifying 15 clusters, and then analyzed proportions of traces in each cluster in each cell type. So all clusters were represented in both cell types. So all these topologies were sampled by both cell types, but the frequency differed. So for example, clusters 1 and 2 were more frequent in human embryonic stem cells, whereas clusters 8 or 9 were more frequent in CNCCs. And then when we looked at these distance maps from all traces in each cluster, we observed that clusters that are relatively enriched in human embryonic stem cells had features that resembled average contact maps from embryonic stem cells, whereas the clusters relatively enriched in CNCCs had features of contact maps from CNCC, including more uniform interactions and the presence of the stripes. So from these observations, we conclude that distinct ensemble average architectures arise not from the presence of strictly unique cell type conformations, but rather through cell type specific changes in frequency of topologies that are commonly sampled by both of these cell types. But given our interest in how precise regulation of SOX9 gene is achieved in CNCCs, we then focused on the feature that was relatively most enriched in CNCCs, and this is this presence of the two architectural stripes. So then we ask what underlies formation of these stripes on 3D contact maps, and are they relevant for gene expression regulation? So previous work from other labs like Leonid Mirny's, Lieberman Eidem, and others suggested that the architectural stripes on contact maps are the consequence of the process of loop extrusion by cohesin and form at the convergent CTCF site where this loop extrusion process is stopped. And the bound CTCF sites are thought to provide a strong barrier to loop extrusion. So when we examined CTCF binding across the locus, we observed that both E1.35 element and the region at the promoter, right upstream from the promoter, were bound by CTCF and bound by cohesin. So RAD21 is a cohesin complex subunit. And furthermore, for aficionados, those two CTCF sites were arranged in the convergent orientation. So then we ask if the mutation of the single CTCF site at the E1.35 element is sufficient to recapitulate effect of this element on SOX9 expression. So indeed, mutation of the single transcription factor site reduced SOX9 expression by about 20%, and this is commensurate with the effect we saw upon deletion of the entire element. And similarly, when we mutated CTCF site right upstream from the promoter, we also saw about 20% reduction in SOX9 expression. So again, while you might think that these are subtle changes, I just want to remind you that at this locus, this is already sufficient to result in phenotypic impact. So then we asked if the formation of architectural stripes on contact maps is CTCF dependent. And indeed, mutation of CTCF site at the E1.35 element result in complete loss of the stripe, corresponding TAL internal stripe. And the mutation of the CTCF site at the promoter resulted in weakening, but not loss of the stripe. And this weakening is particularly evident at the subtraction maps, where you see this decreased interactions across the stripe. But the fact that this stripe did not completely disappear suggests that there is a redundant mechanism that may be stopping loop extrusion at the promoter. But this was not the most striking result from this data. The most striking result is that the contacts across the entire domain were broadly diminished by the mutations of the single CTCF site. So as you can see, there's a lot of blue at this subtraction maps from the mutant CTCF site versus wild type CNCCs. So we were really wondering how the mutation of the single CTCF sites can very broadly affect contacts across the entire domain. And one potential explanation for this unexpected result is that perhaps these two elements are situated centrally within the 3D domain structure and thus pull many other regions inside the topologically associated domain and promote domain compaction. So they are not in the center of the domain in terms of genomic location, but are they in the center of the domain in terms of 3D geometry? So using this individual chromatin traces, Liang Fu calculated median distance from the center of SOX9 TAD across all these other points in the TAD. And the two regions that were clearly located most closely to the geometric center were the promoter and our E1.35 element. And when the E1.35 CTCF site was mutated, the region actually pulled away from the center But at the promoter, it was still retained at the center when the promoter CTCF site was mutated, again, consistent with the existence of the redundant mechanism. So we then calculated mean distance between each region of the TAD and all other points. And again, when we've done that, the E1.35 and the promoter were the closest to all other points, mean distance to all other points within the domain, consistent with the idea that they are centrally located within the domain. And when either CTCF site was mutated, either the promoter or at the E1.35 element, mean distances, not just at these sites, but across the entire domain increased. And this is in agreement with these two sites actually serving, promoting overall domain compactions and increasing the contacts of the entire domain, within the entire domain, consistent also with our subtraction maps. So to recap this part of my talk, at the SOX main locus, we identified two stripe-associated structural elements, which are CTCF dependent. They don't act as an enhancers, but they're instead centrally positioned within the 3D topology of the domain and promote interactions across the domain and domain compactions. They facilitate gene expression and precise SOX9 regulation in CNCCs, but these elements at the same time are distinct from other previously described CTCF elements, such as insulators, because, for example, E1.35 elements facilitates function of long-range enhancers that are both upstream and downstream from the element. So there's no, in contrast to insulators, there's no directionality in which they function. And these elements are also not TAD boundaries. So they're also distinct from the TAD boundary elements, which are also often CTCF dependent. And there are some interesting parallels and distinctions between these elements and tethering elements recently described by Mike Levine's lab in Drosophila, which I would be happy to discuss later. So what is the mechanistic basis underlying these effects? And I already mentioned that formation of the architectural stripes on high-C maps has been previously explained in the context of the loop extrusion process by cohesion with convergent CTCF sites serving as extrusion barriers. And specifically, Casellas and Lieberman-Eiden labs proposed that the formation of these architectural stripes can be explained by the so-called real-in model. And in this model, few assumptions are made that cohesion loading is targeted and occurs near one of the loop extrusion barriers. The next assumption is that cohesion is loaded at low density, such that there's a single cohesion complex extruding between the two barriers at any given time. And the third assumption is that cohesion is highly processive, such that the single cohesion complex can extrude all the way to the next barrier. So when these parameters are incorporated into the polymer models with two loop barriers internal to the domain corresponding to our structural elements, the real-in model indeed recapitulates stripe formation on the ensemble average contact maps. So here's our ORCA data with these two stripes present in CNCCs, and the simulated polymer simulation of the real-in model, you also see the two stripes. However, the real-in model fails to recapitulate contacts when we start looking at the single allele level. So once we move away from the average maps and start looking at individual single allele traces of contacts from one stripe anchor to all points across the domain, and here we sorted the traces based on the most distal interaction from the stripe anchor, then we observed the multiple intervening contact points. You see those little red dots. This between the two that simultaneously contact the stripe anchor, suggesting formation of a multiple loop structure in which multiple distal regions of the domain simultaneously contact the stripe anchor. But this is not at all recapitulated by the real in-model simulations, where the stripe anchor only contacts one point in the domain corresponding to a given position of the extruding cohesin on the DNA. So we then thought we need a new model that really fits the experimental data, and started thinking which assumptions of the real in model are likely to be violated in the context of a large domain such as the SOX9 tag. And one such assumption is that cohesin can extrude all the way between the two barriers in a single loop. And, in the context of our locus, this would mean extruding processively over 1.3 megabases of DNA between the two structural elements. So cohesin is a highly processive enzyme, but recent estimates suggest that in cells, its average processivity is only a few hundred kb, so much less than would be required here. So we knew that one parameter we should adjust in the model is processivity. So we lowered processivity, but to account for this, we allowed for higher density of loading of cohesin. And instead of requiring that cohesin is loaded next to one extrusion barrier, we allowed for random loading across the domain. And sort of the unspoken assumption here is that since now we're allowing for multiple cohesins simultaneously extruding, then they can collide but cannot pass each other. And then we refer to this new model as the multi-loop model. And when we've done polymer simulations incorporating this new model parameters, it much better recapitulated the experimental data from the ORCA, including this intervening contact points between the loop anchor and the furthest contact point. So recapitulates this multi-loop structure. And also very importantly, this new multi-loop model, but not the real in model, recapitulates also central positioning, so central 3D geometry positioning of the two structural elements in our data. So this also suggests that the central positioning is actually, at least in part, a consequence of the active loop extrusion process. So to come back to the original question I posed in my talk, how can this type of mechanism actually facilitate robustness of transcriptional regulation over those ultra long ranges where we have these enhancers that are active well over 1 megabase away? So first, we think that frequent positioning of the promoter at the center of the domain allows for proximity of the promoter to the rest of the domain, including all enhancers, as if the entire domain is now licensed for enhancer activity, and essentially any enhancer active within the domain will be able to act on the promoter. And we also think that multiple of stochastic loading or unloading of cohesin, and then also potentially from kinetic aspect, imply faster bridging of very distal promoter enhancer pairs compared to a single extruding factor. Okay, and now in the last part of my talk, I want to come back to this idea of how does change in transcription factor dosage result in phenotypic specificity. So through our work on PRO band sequence, we actually realized that the phenotypic specificity in PRS arises from the convergence of two factors. One of this factor is enhancer specificity. So I already told you that PRS locus enhancers actually active specifically in the face. And therefore, when you perturb or delete them, as it's seen in patients' mutations, you actually only affect SOX9 function or SOX9 dosage in the face, but not in other tissues and organs in which SOX9 plays important roles. However, both enhancers, the PRS locus and SOX9, are broadly active throughout the face. So whereas the disease manifests preferentially in the lower jaw, raising a question, where is the missing component of specificity? And we actually showed through in vivo modeling in the mouse that the second component of specificity actually comes from the preferential sensitivity of the lower jaw to changes in SOX9 dosage. So with which raises a question, how change in SOX9 dosage can result in phenotypic specificity. So just to sort of recap what is known about dosage sensitivity for SOX9 in the face, loss of SOX9 in mouse knockouts results in broad craniofacial phenotypes and embryonic lethality. 30 to 50% reduction in the dosage, either through heterozygous loss of function mutations, as seen in campomelic dysplasia, or in isolated PRO-band sequence associated with loss of CNCC enhancers, as I have just shown you, results in very specific craniofacial disorder characterized by lower jaw hypoplasia. And then even more subtle changes in SOX9 dosage are associated with normal range facial variation in GWAS studies. And I already mentioned Hannah's result that in mice, 10 to 15% reduction in SOX9 expression during mouse craniofacial development leads to subtle but reproducible changes in lower jaw shape. So we really wondered, how are such small changes in transcription factor dosage interpreted at the molecular and cellular levels, and how do they give rise to phenotypic specificity in the disease, and also in normal range facial variation? So to address this question, Sahin Naqvi in my lab set out to precisely modulate SOX9 protein levels in CNCCs. So briefly, he endogenously tagged SOX9 gene in human embryonic stem cells using FKBP degron and fluorescent tag amnion green, and then differentiated the cells to CNCCs, and then added DTAC, which is a bifunctional molecule that can bridge the FKBP degron and then E3 ligase, resulting in protein degradation. So this system was developed by Nathaniel Gray's lab. But we found it to be highly titratable, dependent on the DTAC concentration, allowing for precise modulation of transcription factor levels. So it's shown here. When we quantified SOX9 levels in single cells by amnion green fluorescence, we observed a unimodal distribution that shifts towards lower levels with higher DTAC concentrations. And these effects are quantified here on the right and presented as SOX9 dosage, percentage of the wild type dose. And of course, the dose here refers to the protein dose rather than the gene dose. So with this system in hand, then Sahin set out to characterize transcription factor dose to phenotype curves at multiple levels of scale, from the chromatin states, gene expressions through cellular phenotypes, and effects on morphology. So first, to assess effect of SOX9 dosage changes on chromatin accessibility and gene expression, Sahin performed ataxic analysis and RNA-seq from CNCCs, which he treated with his different DTAC concentrations, allowing for this variation in SOX9 dosage. And from these results, he identified regulatory elements that were dependent on SOX9. There were about 35 thousands of these elements that are mostly distal. So for simplicity, I will just refer to them as SOX9 dependent enhancers. And similarly, he defined also SOX9 dependent genes. There were over 1,000 of such genes. And both of genes and regulatory elements were defined as those that significantly change their either accessibility or expression when SOX9 was completely degraded. So when there is no SOX9, these elements and these genes are dependent on it. And then for each of these dependent elements, he then characterized SOX9 dosage response curve and fit it with two simple equations, either linear equation or the Hill equation. And he observed that most enhancers and genes actually respond non-linearly to dosage and are better fit by the Hill equation. And from the Hill equation, two parameters can be extracted. One is the ED50 and the other is Hill exponent. And for the purpose of this talk, I want to bring your attention to the ED50, which is defined as the SOX9 dosage at which the enhancer or gene reaches half of its maximal openness or expression. And you can think of this ED50 parameter as a quantitative measure of dosage sensitivity, with the higher ED50 values indicating higher sensitivity to perturbation in SOX9 dosage. So just to give you a couple of specific examples of dosage responses, here's a curve showing changes in hypersensitivity of a specific regulatory elements, and which is actually a typical response that we see for most 80 or 90% of SOX9 dependent enhancers. And this response, as you can appreciate, is not only highly nonlinear, but also buffered against small changes in SOX9 dosage. So this enhancer doesn't care. Its hypersensitivity doesn't change almost at all until you lower the dose below 40% of the wild type dose. So it's really buffered against small changes in dosage up to 50%. And in fact, such buffering would be expected in a robust regulatory system. However, a smaller subset of enhancers and genes show high sensitivity to changes in SOX9 dosage, which with significant loss of accessibility or expression, even at small dosage perturbations at 50% perturbation or even smaller perturbation to SOX9 expression. And we found that the sensitive enhancers are enriched for specific DNA sequence features, and which I'd be happy to discuss later, and that enhancer responses partially at least predict gene responses, meaning that the more and the stronger the sensitive enhancers you have in the vicinity of the gene, the more likely this gene itself is going to be sensitive to SOX9 dosage. So in other words, most of the enhancers and genes that are dependent on SOX9s are highly buffered against small changes in dosage, but there is a subset corresponding to roughly 10% of elements that is highly sensitive. So now the question is, what cellular processes are these SOX9 dosage sensitive genes involved in? And early on, Sahin noticed that multiple prochondrogenic genes are among the dosage sensitive genes. And this was interesting because I already told you SOX9 is required for entry of CNCC intrachondrogenesis, and is also a master regulator of chondrogenesis program. So to characterize this association more systematically, Sahin stratified SOX9 responsive genes into those that are associated with cartilage development or not. And within each group, he further divided them based on whether the expression was downregulated, upregulated, or unchanged during differentiation of CNCC to chondrocytes. And he observed that genes that are involved in both cartilage development and upregulated during differentiation of CNCCs to chondrocytes have substantially higher ED50 values, indicating increased sensitivity to SOX9 dosage. And I will refer to those genes as the prochondrogenic genes. So we then ask if the process of chondrogenesis itself is sensitized to SOX9 dosage. And Sahin modulated SOX9 dosage during differentiation of CNCC to chondrocytes, and then measured mature cartilage formation by colorimetric assay. And he observed that although median dosage curve of cartilage formation was nonlinear, it much more closely the curve of those sensitive prochondrogenic genes as compared to other genes or other regulatory elements which were more buffered in their responses. So in vitro chondrogenesis appears to be sensitized to SOX9 dosage, and this is likely due to increased sensitivity of important prochondrogenic genes. So we next ask if we can link these dosage responses and changes in morphology, first in the context of craniofacial disease, and then in the context of the normal range facial variation. So first in the context of Mendelian disease. So Sahin took all SOX9 dependent or SOX9 regulated genes and divided them into 4 groups based on their associations with Mendelian craniofacial disease. In the first group were genes associated with diseases showing phenotypic overlap with Pierre Robin sequence, namely lower jaw hypoplasia. The next 2 groups included genes that are associated with craniofacial disease, but with no phenotypic overlap with PRS and with either recessive or dominant inheritance. And in the last group were SOX9 regulated genes that are not associated with craniofacial disease. And he then looked at ED50 values within each group, and we observed that genes that are associated with PRS-like phenotypes, or the purple genes, had the highest ED50 values and were thus most sensitive to SOX9 dosage. But interestingly, the most buffered were those green genes, which had craniofacial phenotypes, but unrelated to PRS, and also had dominant inheritance, which may be indicative of dosage sensitivity. So together, this suggests that the SOX9-dependent dosage-sensitive craniofacial genes are generally buffered against changes in SOX9 dosage. However, those that act on the same phenotypic axis as SOX9 are most sensitive. So we next wanted to assess whether similar principles apply to common variation in facial shape in healthy individuals. And to investigate this, we worked with our longstanding collaborator, Peter Claes, and 2 graduate students in his lab. And Peter and his team compared 3-dimensional facial scans from healthy controls and PRS patients to define shape changes along the healthy to PRS axis. And this is also known as an endophenotype. And he then scored over 8,000 healthy European ancestry individuals for this endophenotype and conducted GWAS analysis. This led to identification of over 20 genome-wide significant loci. And as a reality check, the non-coding variants at the SOX9 locus came up strongly in this analysis, but there were also many other loci. So then Sahin looked at SOX9-dependent enhancers that were in the genetic linkage with the endophenotype SNPs, and those again showed higher ED50 values than SOX9-dependent enhancers that were in linkage with facial shape GWAS SNPs that are associated with unrelated normal range facial phenotypes. So just to summarize and finish off my talk, using SOX9 as a model transcription factor and precisely titrating its level at physiological ranges, we found that most enhancers or genes are buffered against small to moderate changes in transcription factor dosage. However, a small subset of enhancers and genes is highly sensitive to changes in dosage. And among the highly dosage sensitive genes, even a smaller subset are effector genes for a specific cellular process, in our case, chondrogenesis. And we propose that these dosage-sensitive effector genes, these prochondrogenic genes, form a regulatory bottleneck, which then transmits quantitative changes in transcription factor dosage to specific changes in cellular behaviors, in turn resulting in phenotypic specificity. So small changes in SOX9 dosage only affect a small number of most sensitive effector genes, leading to subtle normal range changes in phenotype along the healthy PRS axis. Then at 50 to 70% of the wild type dose, the effector genes become more strongly affected, exacerbating the phenotypic impact and leading to PRS, a disease with highly specific manifestations in the face. However, importantly, other key craniofacial genes that are regulated by SOX9 remain robust to perturbation. And finally, only when we lose SOX9 completely, almost completely, this leads to broad effects on many craniofacial genes regulated by SOX9, including those with PRS unrelated phenotypes, which in turn results in broad craniofacial defects and embryonic lethality as observed in SOX9 knockout mice. So I'm going to skip this because of, in the interest of time, and I just want to end by thanking my current and former lab members, and especially those who led this work, Hannah Long, who started our adventure with SOX9, and she's now a group leader at MRC at the University of Edinburgh. Liang Fu Chen, who took on the ORCA project, and we worked closely with Alistair Buttiker at Stanford, and Sahin Naqvi, who led the transcription factor dosage project, and he was course supervised with Jonathan Pritchard at computational biologist at Stanford. And I also want to mention my mentor, David Alice, who unfortunately passed away this year, and he's greatly missed. Thank you very much. All right.
All right. Thank you, Dr. Wysocka, for a really wonderful talk. I'm looking at I don't see any questions in the Q&A just yet. I guess I'll just ask, what are your thoughts just in general on human craniofacial variation, and why is so much variation tolerated, and is there any selection against particular traits?
So I'm sure there is, clearly there is a sexual selection on facial shape. One can argue, one can sort of wonder, is there variation also, is there selection for variation in part because we actually also use face for individual identity and also recognizing individuals within the group and from outside the group. So one would sort of think if this could have some interesting evolutionary benefits, across the vertebrates. Well, humans are not unique in a sense that obviously we don't have much measurements of individual variations in other animals. So the question is there individual variation, let's say, in other species comparable to those of humans? And perhaps yes, and perhaps not. There's clearly a lot of individual variation even in rodents. But whether or not it's comparable to humans is an interesting question, because perhaps species that use facial recognition for individual recognition and communication, perhaps they actually select for higher variation. But nevertheless, if you look between species, there's humongous variation in facial shape. If you think about anteaters or even Darwin's finches, the adaptations to ecological niches that go through face are incredible. So it could be that it is a very plastic structure, and which with a lot of variation, but that of course may carry consequences for disease.
Right. Okay.
So Natalie, I saw there was one. Well, you probably have 30 more seconds. Can we fit one more question in? It just came in from Kimbra.
Oh, okay. Are there environmental influences on SOX9 in humans? It has been identified in temperature-dependent sex determination in reptiles.
Yeah, indeed. And so we don't know if actually for SOX9 at this point, it's hard to prove that the environmental influence in PRS patient, because the PRS patients are obviously they have different mutations which can affect gene expression to different extent, but there's also phenotypic variability that perhaps cannot be fully explained by the genotype, and I suspect that this results from environmental influence on the locus regulation. So I guess we probably now would have mouse models that we could more systematically investigate that under conditions where what's the genotype convergent between the genetic and environmental influences. When there's a small perturbation in SOX9 expression added with an environmental insult. And it is known that environmental influences, for example, influence prevalence of clefting. So wouldn't be a huge surprise that there's an environmental component on variation.
Well, Natalie, I think we better wind things up. We're running over 1 o'clock.
I'm sorry, I somehow
No.
Absolutely fascinating talk. And I'll just in the last 10 seconds here, just thank you, Joanna, for the spectacular talk, and just reinforcing the importance of all of those nucleotide sequence variations that occur between the coding sequences of genes. So we've got to do whole genome sequencing if we're ever ultimately going to understand all of the human variation. And that happens to be an area not only am I personally interested in, but also an area that we're very interested in here at NIEHS. So thank you again for spending some virtual time with us, and hopefully at some point in the future, I know that many of my colleagues would love to have you here in person. So we'll follow up.
Thank you very much for hosting me. And now I'm meeting with the students, right? So I am switching to okay. students.
Sounds
in
good.
Canada.
Okay, take
Okay,
care.
thank you.
Okay.
Bye.
Thank you. And Natalie, I'll see you later.
Okay, sounds good.
Take care.
Okay, bye-bye.